Hello everyone, welcome back to Med with K. Today we'll be talking about glycolysis. So this topic was suggested to me by one of you in the comments. So before we proceed with this topic, I would say that I have already made a video on how to approach anatomy, then specifically another video for gross anatomy, then a video on physiology, how to approach physiology, and a one on biochemistry also. So I would really like if you guys see those videos, it would really add value. Now, coming to this topic, this is glycolysis. So, we have discussed in one of our videos about the medical terminologies that is, glyc means glucose. Okay, this is glucose. Lysis means breakdown. So, if I talk about glycolysis, it is basically breaking down glucose into something. Now, that something is pyruvate or indirectly it can be lactate. So moving further, this process of glycolysis, it takes place in the cytosol. Cytosol or basically the cytoplasm. It is a 10-step reaction and it is of two types, that is aerobic or anaerobic. So the one in which pyruvate was being generated, it is the aerobic one and lactate is anaerobic one. So in aerobic, the end product is 7 ATP plus 2 pyruvate, and in anaerobic, we get to ATP plus 2 lactate plus 2 NAD plus. Now, this reaction basically shows the anaerobic type of glycolysis. The features of glycolysis. So these are the salient features. It occurs in cytosol of all the cells. Glycolysis is essential for the brain because brain is highly dependent on glucose for energy. Then it is the only source of energy for WBC, RBC, cornea, lens, and white muscle fibers. Now, why is it the only source of energy? Because these cells, they lack mitochondria. So, if they do not have the mitochondria, which is the powerhouse of the cell, they cannot generate their own ATP, and thus they have to be dependent on the process of glycolysis. So, and another important is that this 10 series reactions, it has 3 irreversible reactions. Now, again, you guys know that I want to focus on the basics and concepts. So before we move to the glycolysis pathway, glycolysis reactions, I would really want you to understand that how glucose is transported and what do you mean by the phosphorylation and all? These are very important stuffs, like the role of insulin and then the role of glucagon. So coming to glucose transport. So glucose is transported via two methods. One is sodium ATP independent and another is sodium ATP dependent. So what do you mean by sodium ATP independent? That it means that for the glucose to come into the cell, it does not require the help of sodium. And for the one on which it is dependent, it requires the help of sodium. So see, this is your cytosol. Here glycolysis takes place, right? This is your cell membrane and this is your ECM. So glucose, it comes from the ECM. Then we have a glucose transporter, right? So we call them GLUT, G-L-U-T, glucose transporter. So it is of various types. We'll discuss this over here. So this glucose transporter, it is a uniport. Uniport is one-time movement, right? So this. So this glucose comes in and goes into the cytosol. Okay. Now, some important questions, MCQ questions, PG questions, and more importantly for your concepts. GLUT1 is in RBC and blood-brain barrier. GLUT3 is in neurons. GLUT4 is in muscles and adipose tissues. So the role of GLUT1, 3 and 4 is to uptake glucose from the blood. Now coming to GLUT2, GLUT2 is found in liver, kidneys and pancreatic beta cells. So it transports glucose into the cells when blood glucose is high, okay? Or transports glucose from these cells when blood glucose is low. That is in fed state. What does it do? ये liver है. This is your blood. It transports glucose from the blood to the liver in well-fed state. And in fasting state, it does the opposite. GLUT5 is not for glucose, it is for fructose and it is found in small intestine and testes. It is another important question. Now coming to sodium and ATP dependent. So there's a saying that wherever sodium goes, water goes and glucose also follows, right? So you see, sodium is going inside and then glucose, galactose, they also follow it, right? Now coming to another important concept, that is of insulin and glucagon. So insulin, when you hear the word insulin, automatically two terms should come to your mind, that is fed state and dephosphorylation. So whenever we are full, our body releases insulin. Now this insulin, it activates phosphatases. The ACE is coming, right? ACE means removing something, breakdown of something. So it removes the phosphate groups and leads to dephosphorylation. Now, what does insulin do? Insulin, it enhances the uptake of glucose. Basically, to use glucose, to store glucose, it is an anabolic reaction and it inhibits glucose production, right? So, why does it inhibit glucose production? Because insulin release होता है. Insulin काम क्या है? इंसुलिन क्या करवाता है? वो सेल्स में ग्लूकोज का अपटेक करवाता है. So it would not want the body to make more glucose, right? It would want that the cells, they uptake glucose and then they utilize it for their needs. So glycolysis, the method is glycolysis, wherein the glucose is being broken down into something and it is being utilized by our body. So glycolysis is mainly depends on dephosphorylation. The enzymes of glycolytic are activated in dephosphorylation state. Am I clear with it? So glycolytic enzymes are activated in dephosphorylated state. Why? Because of insulin. Now coming to glucagon. So glucagon is another hormone which is very essential. So with insulin we had fed state and dephosphorylation. With glucagon we have starvation. So what does glucagon do? Glucagon in liver, it binds to GPCR receptors. Then it activates GS protein, which activates adenylate cyclase. So this pathway is important. Now this adenylate cyclase, it converts ATP to CAMP. Now this CAMP, it converts inactivated protein kinase to activated protein kinase. This activated protein kinase, it helps in the phosphorylation of protein kinase. So glucagon is related to phosphorylation. Now what does glucagon do? Glucagon, it breaks glycogen, it enhances gluconeogenesis, right? So insulin में what happened? Insulin में the cells wanted glucose, right? Cells wanted glucose because बहुत सारा glucose था blood में. Insulin में there was high blood sugar and the cells wanted to use it. But in glucagon, we want the blood sugar to be high. We want to prevent the state of hypoglycemia. So for that, we need to do glycogenolysis, that is breakdown of glycogen, or gluconeogenesis. And we need to stop glycolysis, right? So glucagon activates pathways that make glucose or save glucose. These pathways, they happen to be switched on by phosphorylation. So fasting enzymes are responsive to phosphorylation. So in crux, you have to just remember two things. Insulin का main काम क्या है? Insulin, blood sugar is high, insulin is released, these sugar is transported to the cells. Glucagon, it stimulates production of glucose. Correct? Glucose production. Now coming to glycolysis. So in glycolytic reactions, we have three phases. One is the energy investment phase, one is the splitting phase, and one is the energy generation phase. Now in energy investment phase, 2 ATP is converted into 2 ADP. Splitting phase, nothing takes place. And in energy generation, 4 ADP is converted to 4 ATP and 2 NAD plus is converted to 2 NADH. Now about this you will get to know आगे. Now coming to net. So net, there's a gain of 2 ATP and 2 NADH. Now 1 NADH is 2.5 ATP. So 2 NADH would be 5 ATP. 5 plus 2, 7. So total ATP is 7. Now coming to energy investment phase in detail. Coming to energy investment phase. So it is a 3-step reaction. Number 1, glucose is converted into glucose 6-phosphate. The glucose is a 6-carbon compound. It is converted to glucose 6-phosphate. So over here, one energy step takes place, that is ATP is converted to ADP. So this is an energy investment phase. The enzyme here is hexokinase or glucokinase. And magnesium plus 2 is also there. Now, you can have a question that what is the difference between hexokinase and glucokinase? It is a very important one. We'll discuss it. Now, this glucose 6-phosphate, it is converted to fructose 6-phosphate by an enzyme isomerase. So, this is a reversible reaction. So, this was irreversible, then this was reversible, and this reaction that we'll be talking about is irreversible again, right? So this fructose 6-phosphate, it is converted to fructose 1,6-bisphosphate, and again an energy, that is ATP, is utilized. The enzyme here is phosphofructokinase 1. Phosphofructokinase 1. So this is a very important step. It is irreversible, it is the regulatory step, and it is the rate limiting step. Now coming to each one in detail. So this step is very crucial. Why? Why is the phosphorylation important? So the answer lies here. The phosphorylated sugar molecules, they do not readily penetrate cell membranes because there are no specific transmembrane carriers for these compounds. So what happens is whenever phosphorylation occurs, so phosphorylation, they cannot enter the cell membrane, right? They cannot enter the cell membrane because they are way too polar to diffuse through the lipid membranes. So this is the irreversible phosphorylation of glucose. So therefore, it effectively traps the sugar as cytosolic glucose 6-phosphate and commits to further metabolism in the cell. Basically, we do not want the glucose to be utilized by the cell. Therefore, we have to convert it into such a thing that it cannot be absorbed into the cell membrane. That's why this step is very important. Now, if I talk about glucokinase or hexokinase. So, glucokinase is also known as hexokinase 4. So, this table is very important. So, one exam tip, whenever a question on glycolysis comes, a long question, and if you are talking about this part, make sure that you mention the difference between hexokinase and glucokinase. It will fetch you some extra marks and will leave you a good impression. Okay, so coming to hexokinase and glucokinase. The hexokinase is found in tissues, glucokinase is found in liver and pancreatic cells. Glucokinase acts when the blood glucose is greater than 100 milligrams per deciliter. That means its affinity for glucose is low. So if the affinity is low, then Km is high. And hexokinase, it can be used in low concentration, therefore giving a higher affinity, and Km is also low. Now what happens is this hexokinase, it is not inhibited by glucose 6-phosphate, but hexokinase 4 is not inhibited by glucose 6-phosphate, but hexokinase is inhibited by glucose 6-phosphate. Now what happens is this see, hexokinase is activated when blood sugar level is very less, right? So when the blood sugar level is very less, we do not want glucose to be converted into pyruvate or lactate. We want this glucose to be supplied to the brain. So that's why this glucose 6-phosphate, it inhibits hexokinase because it can act on very low concentration of glucose. Therefore, prevents overuse of glucose. So this was the same thing that we discussed. Now coming to phosphofructokinase. Phosphofructokinase 1, it helps in the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. Now what happens is ATP and citrate, they inhibit this enzyme, and AMP and fructose 2,6-bisphosphate, they activate this enzyme. Let us talk about fructose 2,6-bisphosphate. So what happens is this fructose 6-phosphate, it can convert into fructose 2,6-bisphosphate by the help of phosphofructokinase 2. We have discussed about PFK1. So about PFK2, it is a bifunctional protein that it can exist either as a kinase or as a phosphatase. So what happens so the question is, how does this activate this, right? So what happens? Glycolysis, it is taking place in well-fed state. Well-fed state, that is insulin, right? So what does insulin do? Dephosphorylates. So insulin, it dephosphorylates phosphofructokinase 2, which leads to the formation of fructose 2,6-bisphosphate. So now when fructose 2,6-bisphosphate is increased, it goes and it activates PFK1, which leads to fructose 1,6-bisphosphate formation, which leads to glycolysis. Now a question. So what happens during the fasting state. So in fasting state, what happens? This phosphofructokinase 2, it acts as a phosphatase, right? So because of the influence of glucagon. So what happens? This fructose 2,6 bisphosphate, it is converted to fructose 6 phosphate, and through this it takes part in gluconeogenesis. Then it doesn't come and participate in glycolysis. Okay, so one more point, that phosphofructokinase 2, in its kinase form it is known as PFK2 only, and the phosphatase form it is known as fructose 2,6 bisphosphatase or phosphorylated PFK2. So this was a very important concept. Let's move forward. Coming to the splitting phase, so fructose 1,6 bisphosphate, it splits into G3P, that is glycerol 3-phosphate, and DHAP, right? So this 6-carbon compound, it splits into two compounds of 3-carbon compound, and the enzyme is aldolase. Now what happens is, for the process of glycolysis, DHAP, it converts to G3P via an enzyme isomerase. So aldolase B is found. So this is aldolase. Another important thing is there's an enzyme aldolase B. It is found in fructose metabolism. And for glycolysis to continue, DHAP must be converted to G3P. So one molecule of glucose will give two molecules of G3P. Coming to energy generation phase, so G3P is converted to 1,3-bisphosphoglycerate, and here 2 NAD plus is converted to 2 NADH, right? So remember this point, it is energy generation. So iodoacetate, it inhibits this enzyme, thus it inhibits the process also of glycolysis. Now this 1,3-bisphosphoglycerate, it is converted to 3-phosphoglycerate, and the enzyme is phosphoglycerate kinase. So an important thing to remember about this enzyme is the physiology, is that it is physiologically reversible kinase. And arsenate, it inhibits this enzyme. So in cases of arsenic poisoning, glycolysis won't continue because this step would be inhibited. Now what happens in a lot of cases is that 1,3-bisphosphoglycerate, it gets converted to 2,3-bisphosphoglycerate, which gets indirectly converted to 3-phosphoglycerate. So don't be confused. This is known as the Rapaport Leuberg shunt or 2,3-BPG shunt. So the need of this enzyme is, what does it do? It is present in high concentration in RBCs and serves to increase the oxygen delivery. So without this enzyme, the hemoglobin, it will hold on to the oxygen and it won't deliver the oxygen to the tissues, leading to hypoxia. So we want this enzyme to be there because it will help in the unloading of oxygen. So now the question is, what is more important, to prevent hypoxia or to generate ATP? So of course, more important is to prevent hypoxia. Therefore, in some cases, RBCs, they choose this pathway and they let go of ATP generation because of priorities again. Now this 3-phosphoglycerate, it is converted to 2-phosphoglycerate. Again, the enzyme is a mutase. This 2-phosphoglycerate, it gets converted to PEP, that is phosphoenol pyruvate, and enzyme is enolase. Again, an important thing is fluoride inhibits enolase. Now this PEP, it gets converted to pyruvate by the help of pyruvate kinase. Now again, this pyruvate kinase, you tell me when will it be activated? It will be activated in dephosphorylated state. Why? Because it is a glycolytic enzyme and it will be inhibited in phosphorylated state. So again, two ATPs form. So this part, when ADP forms ATP, is known as substrate level phosphorylation. So we have seen it over here also, substrate level phosphorylation. So again a question comes that how many ATP are generated from substrate level phosphorylation and glycolysis? The answer is 4 ATP. 2 ATP, 2 ATP. Okay. So this was all about aerobic glycolysis. Now, if we have to talk about the anaerobic glycolysis, you will see that this PEP is converted to pyruvate. Okay, we saw that. Now, this pyruvate is it in its enol form. So it is converted into its keto form, right? This pyruvate was it in its enol form, it gets converted to keto form. Now what happens is over here 2NADH gets converted to 2NAD plus and this lactate is formed. So the end product is 2 ATP plus 2 NAD plus is formed, right? Now, if I talk about pyruvate kinase, it is activated by fructose 1,6-bisphosphate. This is known as feed forward mechanism, and it is inhibited by ATP. So, we discussed the earlier that this fructose 1,6-bisphosphate, it activates pyruvate kinase. Okay. Now coming to covalent regulation. So what happens is in state of hypoglycemia, that is blood sugar is low. So glucagon is activated, and again we have discussed that it leads to phosphorylation of pyruvate kinase of a protein kinase which inhibits pyruvate kinase because pyruvate kinase is a glycolytic enzyme and it is activated in dephosphorylated state. So when phosphorylation occurs, it becomes inhibited or deactivated, thus leads to decrease of glycolysis. Now talking about regulation, we have discussed it already. First step was of glucokinase or hexokinase, then there was a step of phosphofructokinase and then a step of pyruvate kinase. So if the regulation comes as a question, you have to mention this step, you have to write this slide, then you have to talk about PFK I because it is a rate limiting step, and then you have to show this part of the pyruvate kinase, this slide. Okay, so this would be your regulation. Now net ATP production from glycolysis is zero in RBCs. In which conditions? In arsenic poisoning and in Rapaport Leuberg shunt. Now coming to defects of glycolysis. So because glycolysis, it mainly affects the RBCs because RBCs rely only on glycolysis. So defect would lead to decrease in ATP and thus would lead to hemolytic anemia. So if there's a deficiency in pyruvate kinase or there's a defect of pyruvate kinase, it can lead to chronic hemolytic anemia. It can lead to increased 2,3-BPG. So why increase 2,3-BPG if pyruvate kinase is not there, right? So this step, if pyruvate kinase is not there, then uh, phosphanol pyruvate would be more. So if we go backwards, this would be more, this would be more, this would be more, and thus 2,3-BPG would be more, right? So if 2,3-BPG is more, then what would happen? No tissue hypoxia. And similarly, in phosphofructokinase 1 deficiency, it is also known as Torrey disease. It is a glycogen storage disorder type 7. So over here, decrease 2,3-BPG. Now, why decrease 2,3-BPG? Because you see, it inhibits. So this step would be inhibited. If this is not being formed, then how can you expect 2,3-BPG, which is much later, to be formed, right? So over here we can experience tissue hypoxia and exercise intolerance. So this was it with glycolysis. And again, an important part is the fate of pyruvate. So you see glucose से pyruvate बनता है via glycolysis. Alanine with the help of alanine transaminase can lead to the formation of pyruvate. Same with lactate in the process of gluconeogenesis. Then this pyruvate, from it, ethanol can be made in yeast and lactate can be made in humans. So this is the fate of pyruvate. You can have a look. So in short, if I were to summarize it, so in dephosphorylated state, insulin is the hormone. In phosphorylated state, glucagon is the hormone. Over here in dephosphorylated state, pyruvate kinase and phosphofructokinase 2 is activated. And over here, PFK, over here, pyruvate kinase is inhibited and fructose 2,6-bisphosphatase is activated. So glycolysis is on in dephosphorylated state and in phosphorylated it is off because over here we have to use excess glucose and we have to spare the glucose for brain. So that was it for the video. Here are my notes. If you want them, you can tell me. I can put it in the comment section. And that's all. I hope you liked the video. I hope you gained something out of it. You can comment be the next topic that you want me to discuss. Till then, have a great day. If you like the content, please like, share, and subscribe. That would mean a lot. Thank you so much.